Okay, so this is the last installment of your favorite show, Game of Clones and Immunology. Um, we're going to finish up hypersensitivity today and type 3 and type 4. This won't take too long, so it shouldn't be too much. So for type 3 hypersensitivity, these are this results from immune complexes formed from complement. And you all know about complement. You should know about complement. Remember, with the MAC attack complex and C1Q, C3, C3b, and all of that. So, large immune complexes can be formed from complement. You can have off-target um, activation, and these things can be deposited in the blood vessel walls or in tissues. And when you have activation of complement, as you know, you can get histamine release or mast cell actually activation, histamine release, and so those will attract neutrophils and lead to inflammation at those sites. Now, for example, if we have a bacteria, uh, there's a bacterial complex that forms a film or a layer of bacteria on um, on the tissues. These can attack and have bystander effect. You can get then activation of complement, which then leads to activation of complement onto bystander cells and activation of inflammatory response. Now, if so, this can occur particularly if you have, let's say that you have for um, an allergen that is large and complex. If you get um, formation of aggregates of that antigen with antibody, you can get activation of complement in that area, and that activation can lead to MAC forming, um, MAC attack, or MAC formation on normal tissues. You just sort of just get such an overwhelming amount, it overcomes the ability for the cells to um, shield themselves from the complement activation. And of course, this is going to attract neutrophils and eosinophils attract into that area to lead to potential tissue damage. Um, there are a number of sicknesses that are result from this type of interaction. Serum sickness, where you have blood that's has been has attacked by complement. Pools in the can be pools in the feet, are the arthritis reaction, rheumatic fever, rheumatoid arthritis, farmers and pigeons' lungs. Anything where you breathe in a lot of allergens, for example, can lead to that. So there are a number of examples of conditions that can lead to this reaction. And you can see here, this is another picture in which you see, for example, if you have a small antigen or antibody complex in the epitopes, for example, this could be pollen in which you have epitopes, um, the same epitope along on the particle. You can get um, aggregation, in which aggregation, because your antibody is bivalent, IgG is bivalent, and so you get aggregation um, across that complex, and this can activate C1Q, and C1Q can then activate the deposition of C3b, C4b, C2b, C3b activation, and this can lead then to then um, histamine, and these complexes are huge. So what actually ends up happening is they can precipitate out in the glomerular, the kidneys. They can precipitate out in small blood vessels. They can precipitate out, for example, in areas where you have low your blood is flowing very slowly. For example, in the feet, where it's taking a long time, often is a case. There is a way to get rid of these. You do have a way of getting rid of these, and that is erythrocytes actually carry receptors on their surface called CR1 receptors, which will bind to C3b, and they can bind to any C3b that's been activated and is binding in on these complexes, and basically deposit them on phagocytic cells, like macrophages, which will dispose of them. Now, when you have a locally injected antigen in immune in an immune image in an immune and an antigen in an immune individual with which has IgG antibody, if you have a antigen that's locally injected, then you can get complexation, fairly large complexation of that antigen. Remember, it has to be polyvalent, has to have the same epitope, and that activation can then, of course, um, you can get great deals of cross-linking within there, and then that will activate C1. That by C1q will bind to this, and when it does, it will then lead to activation, particularly degranulation of mast cells, um, which will then lead to local inflammation, um, in which you get perfusion of vasodilation because the histamine is being released, and you basically get a rash and bumps, things like that. And this can happen within an hour to two, 1 to 2 hours. And you can see here, this is a typical kind of reaction in which someone hasn't had an allergy. They've gotten the complex it's been in the blood, or it's been injected. and in this, you can see here, you'll see this in the. This is often typically seen in the feet, because these are areas, of course, of where the blood flow is the slowest, and the feet actually become very itchy and very irritated. I know I've had this reaction when I had a hypersensitivity reaction to a sulfa drug, and once you go off the drug, or your exposure to the allergen goes away, it'll gradually reduce, and because you're getting rid of it. And you can see here on this child, you've got a splotchiness all along the child. And so for inhaled types of allergens, the case like Fommer's lung, it's the alveolar and capillary interface. For subcutaneous, it's the arthros reactions, perivascular area. For intravenous or high dose types of allergens, you get vasculitis, nephritis, or arthritis, in which it's in the site of the complex deposition is in the blood vessel walls or the renal glomerular, which can lead to some kidney damage, and in case of arthritis, to joint space damage. Now, what you generally see is the sort of time period for this is that there's the level in the plasma. So if there's an intravenous injection, say this is a drug molecule, there's an intravenous injection, or it could be a from an insect bite, then there's the concentration goes up, and it will go down over time, and if it's long lived, if it's there for quite some time, if it goes down, you're going to see the antibody in the blood go up with time, and then it's this area here where you hear right here in this middle, in which you see where there's still plenty of antigen around as the antibody is being generated, that you're going to see where antibody antigen complexation can form. Once the antigen is gone, antibody of course will go up to a certain level off, but the reaction no longer can take place because antigen is gone. So it's the sort of window of time in which antigen and antibody are around that you get this reaction. Now, for type 4 delay is type 4. All the other ones that we talked about, 1, 2, and 3, were called immediate hypersensitivity reactions. Type 4 are delayed hypersensitivity reactions, and these are thought to be a response to tissue harboring bacteria or parasites. It occurs most commonly with topical agents which react with the skin. It's called contact dermatitis. such as cosmetics, uh, poison oak, chemicals, um, can be lotions, for example. Anything that I've seen cases in which protein, there was a severe case I saw um, in which a person had it to the clothing. It turns out it wasn't the clothing that was causing this, but it was the detergent that was being used. Detergents are actually, a lot of detergents are full of enzymes to break down the proteins or stains that contain protein as protein-based material, and so that actually comes out on the skin, on the clothes, and because those enzymes are incredibly stable, um, and that can be, and people can be very quite sensitized to that. So they have to have special detergent that doesn't put that on there. Um, the key thing is the development of these Th1 cells, sensitized Th1 cells, which secrete cytokines capable of activating and attracting macrophages into that area. And so, for example, and so typically the way this works is an antigen is applied to the skin or the surface area, will form a complex, covalent complex generally, with proteins on the surface of the cells or in that area in the cell membrane. This gets picked up by an antigen processing cell, and then those antigen processing cells will move into the lymphatics. And this is like a follicular or a this will be a dendritic cell. Uh, uh, those will move into the lymphatics, and when they're in the lymphatics, they go through antigen processing, like we've seen before, and presentation and activation, uh, and particularly to Th1 cells, or to naive Th0 cells. Um, when this activation occurs, of course, these Th1 cells will differentiate into more Th1 cells, including memory cells. But these Th1 cells will then move into the lymphatics and then go into the periphery, and they will interact with antigen processing cells again that are there, and further activate, um, further activate more Th1 cells, and these, of course, will um, um, lead to attraction of macrophages into that area, and then basically inflammation within that area. Um, I don't have it here though, but it is possible for uh, it is possible to have some CTL activity within these areas, or TH or CD8 cells. Um, but for the purposes of right now, we're just going to stick with the Th1 cells, and the main event bringing in macrophages, traction, initiation of cytokine activation, and inflammation at that area. And that's where where you get that skin and that rash happening. And you can see here, this is it can be quite dramatic. This is pentadecacatechol, which is the active ingredient in poison ivy. And you can see here, it's basically a long chain, almost like a lipid, in which you have has several points of unsaturation. This making it very, very lipid soluble, and so easy to get into skin, apply to skin. And then this catechol, these two oxygens here, these kinds of molecules will interact, and bind to just about any protein. They're well known. These kinds of structures are well known to bind and form strong bonds to lots of different proteins. Um, and so these kinds of things, will bind and lock on to the proteins in the skin. And this is the kind of thing this is the kind of reaction you get. This is a pretty severe reaction. Now, I just want to point out, the reason it's called delayed is when you go back, and just go back here a second, is this all takes up quite a bit of time for the antigen processing cells to process the antigen, go into the lymphatics, go to lymph node, go through, find a TH cell, and activate the TH cell, and then TH cell proliferation, differentiation, and back, and the cell has to migrate back in. This all takes quite a bit of time, and so that's why it's called delayed, because unless you've had a prior exposure, it can take a while. And so if the antigen, in lots of cases, the antigen is still there in the skin or it's seeped into the skin, those cells don't go anywhere. And so the antigen is still in that environment and it's not really removed. Um, the allergen is there. And so, but it can take several days for this to all process and to eventually come about. And then, the next exposure, of course, is memory cells. Some of these TH cells are going to be in that area. You've made memory cells that will actually harbor in this area, tissue memory cells, tissue TH cells that will be harbored in this area in case there is a second exposure. But the initial part can be delayed for quite some time. There are ways, therapeutics based on type 4 hypersensitivity. Um, there are a number of diseases that are associated with several conditions such as tuberculosis, leprosy, leishmania, deep fungal infections. In fact, tuberculosis and leprosy are basically um, activation of the immune response to the intracellular mycobacterium, which is growing in that and gets processed and then presented on those cells. You have some antigen that's presented in the surface of the cell, and these type of cells, this kind of interaction leads to overactivation of the immune response, leading to tissue destruction. It's not the mycobacterium that causes the disfiguration of leprosy or the destruction of the lungs, like in tuberculosis. It's actually the overactive immune response that happens, particularly macrophages. So type 4 is also associated with allergic reactions to any contact antigens like poison ivy and poison oak. Now, there are allergen patch test kits. We've seen the test kits before for the other allergens, so you can use these test kits to determine if a person um, has a contact dermatitis. They usually go through these test kits to determine what is likely to be the cause. Lots of times it doesn't work that well because there's so many allergens possible. But it's a way that the allergist can go through and potentially locate them. I've known several people it never worked for them to do that. Other people I've known actually determined and was helpful to find out what allergen in their environment is the major problem, spore or contact, what they're contacting. You can go through a desensitization, oral desensitization. Um, these involve extracts from the actually poison ivy. This is the Latin word for that, and it involves basically ingesting small amounts of the extract over a period of time to again produce basically eventually just maybe IgA to block that interaction, so that you producing IgA, maybe a little bit of IgG, but probably mainly IgA, to block the interaction uh, between the allergen um, and the APCs, to block it and remove it from so that it doesn't activate the immune system. There are some adverse effects you can get. One of my uh, favorite adverse reactions, I know everybody's favorite, is purititis ani, or a burning sensation in your rectum. Occurs briefly during the first weeks, basically because you're getting used to the allergen, which the allergen in case of the poison oak allergen in your along your in your GI tract. Um, Now it's turned out to be this has been adequately potent and as judged by the FDA. And I'd say this is basically can be used for people who have need to be outdoors, are going to be kind of in contact with it quite a lot. This is one way to desensitize a person. It should be noted that the desensitization, any of these desensitizations, is not permanent because, of course, over time, the amount of the antibody in the plasma or antibody that's being produced and excreted in the urinary tract or through sweat is going to drop over time for that particular allergen. So sometimes people do have to go and be resensitized, re-desensitized. In the case of for poison ivy extracts, you don't even have to. That allergen is volatile enough that it can actually is kind of a haze over a poison ivy patch. It's actually volatilized enough that you can pick it up on in the area without even touching the leaves. So some people who are quite incredibly sensitive don't even have to touch them. They can just come within some short distance of it and have the reaction. Now, there's a number of excuse me. Each one of the hypersensitivity reactions we've talked about can induce a drug immunopathology, and these immunopathologies are quite common and are the pathologies that are in some cases the most life-threatening for a lot of drugs. In many cases, the drugs aren't necessarily all that toxic unless taken in very high doses. Excuse me, but but they can potentially induce an immune response, and that immune response at relatively low doses can lead to a systemic anaphylaxis or local anaphylaxis. So each of the four types of hypersensitivity can lead to deleterious effects when a drug is administered. So type 1 would be can cause allergic anaphylactic reactions due to allergen complexation with mast cells. So if you have a self protein in which the protein has several different copies of the drug that's bound to it, that's an allergen because you're polyvalent, same epitope, and the antibody, the IgE, can bind to the drug, which is the epitope, and activate the mast cells. So that can happen. For type 2, you can cause blood cells that are labeled to be removed by complement or ADC. So for a type 2, for the blood cells themselves, as we saw with the fetus there, the blood cells can recognize the drug that labels the red blood cells, and when it does, it will initiate the antibody that's produced from that. The antibody produced will go after those blood cells, and you can have complement activation or you can have ADCC. For type 3 hypersensitivity reactions, these are caused when you have complement complexation, when you have large, and for example, you have IgM, you have these large, enormous complexes that can be formed and precipitate out or be in these tissue spaces, can then lead to activation of complement in those areas, and you get innocent or bystander reaction in tissues which from these complexes. And this would be, for example, in the small blood vessels of the feet, or for example in the kidneys and the glomerular. You can see this, the blood vessels in the kidneys. And then for type 4 is basically anything that can label a drug that potentially reacts with the skin, a lotion, or anything that would label the skin and result in inducing a toxic response from a T cell response. And this, of course, you can have memory. And so in this case, this This doesn't have antibody involved. Actually, all the 1, 2, and 3 all have antibody involved. For type 4, it's all cell based on the um, T helper cells, Th cells being involved and being activated through largely through these Langerhans cells, which are in the skin, which will process the antigen, the drug peptide, the drug protein. So, to close, take home message here is to remind you that one, allergic or hypersensitivity reactions result from a non-desirable immune response. It's not something that we want. We want to have happen. It's something that's built into us, particularly to protect us from a variety of different potential pathogens. Um, Two, both CMI, AMI, and complement are involved in allergic reactions. Um, 1, 2, and 3 type hypersensitivity reactions involve AMI or complement, and all three involve antibody, whereas CMI type 4 is where CMI is involved. And 3, hypersensitivity reactions are a major reason for drug induced pathologies. So both type 1, type 2, type 3, and type 4, if drugs can be involved and activate those 4. Now, when it turns out that um, for most of these drug pathologies, uh, the symptoms are noted, and for example, for type 1, if it appears to be a mast cell activation, particularly if it causes a drug causes a sort of an asthma-like condition, then it's going to be primarily an IgE response. But for most cases, you're not going to necessarily determine what type it is, type 1, 2, and 3. You'll be more concerned with the after effects that are happening and how to deal with them, knowing that histamine is released in these lipid mediators, etc. So for that, it's been a pleasure teaching you this semester. I wish you the best of luck the remaining semester, remaining part of the semester, and I will. Some of this material, just to remember, this material will come up on an exam, uh, and Dr. Probably on the next exam, which Dr. Anderson, which will be coming from Dr. Anderson, and I'll have a little bit to say about that later when we get closer to that. Take care.